Good day. This is the Professor, CPMD PhD. And today's Wednesday, September 13th, 2023. It is 2:52 and 39 seconds, AM Eastern Standard Time. And today's research, AI tools in cancer. So let's jump to it here. AI tool helps optimize antibody medicines. Machine learning points out why antibodies fail to stay on target, finding the molecules that aren't markers of disease and suggest better designs. Date September 11, 2023. Source, University of Michigan. Antibody treatments may be able to activate the immune system to fight diseases like Parkinson's, Alzheimer's, and colorectal cancer, but they are less effective when they bind with themselves and other molecules that aren't markers of disease. They could use the models to pinpoint the positions in antibodies that are causing trouble and change those positions to correct the problems without causing new ones. The models are useful because they can be used on existing antibodies, brand new antibodies in development, and even antibodies that haven't been made yet. Antibodies fight disease by binding specific molecules, called antigens, on disease-causing agents, such as a spike protein on the virus that causes COVID-19. Once bound, the antibody either directly inactivates the harmful viruses or cells or signals the body's immune cells to do so. Unfortunately, antibodies designed to bind their specific antigens very strongly and quickly can also bind non-antigen molecules, which removes the antibodies before they target the disease. As such, antibodies are also prone to binding with other antibodies of the same type and, in the process, forming thick solutions that don't flow easily through the needles that deliver antibody drugs. The ideal antibody should do three things at once: bind tightly to what they are supposed to, repel each other, and ignore other things in the body. An antibody that doesn't check all three boxes is unlikely to become a successful drug, but many clinical stage antibodies can. In their new study, the team measured the activity of 80 clinical stage antibodies in the laboratory and found that 75% of the antibodies interacted with the wrong molecules to one another or both. And by changing the amino acids that comprise an antibody and then turn the antibody 3D structure to prevent antibodies from misbehaving because an antibody structure determines what it can bind. As some changes can cause more problems than they fix, and the average antibody has hundreds of different amino acid positions that could be changed. Exploring all the changes for a single antibody takes about 2 workdays with the models, which is substantially shorter compared to experimentally measuring each modified antibody, which would take months at best. The team's models, which are trained on the experimental data they collected from clinical stage antibodies, can identify how to change antibodies so they check all three boxes with 78% to 88% accuracy. This narrows down the number of antibody changes that chemical and biomedical engineers need to manufacture and test in the laboratory. As some companies have developed antibodies that they are really excited about because they have desired biological activity, but they know they are going to have problems when they try to use these antibodies as drugs. That is where the researchers come in and show them the specific spots in their antibodies that need to be fixed, and they are already helping out some companies to do this. And this is the Professor of Microbiology, Bioinformatics, Pathophysiology, and Teacher of Research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have an awesome day.